Okay everyone, welcome back. We have seen DNA replication, we have seen all the enzyme that helps during the replication process. Okay, today we will see that DNA replication is a semi-conservative. DNA replication is a semi-conservative. A new topic. We have discussed replication of DNA. Now this DNA is said to be semi-conservative. That means semi means partly, okay, partly. Conservative means old. Partly, conservative means old. Okay, that means the strain, that means the new strain which was synthesized, that new strain is synthesized from the old strain of DNA, and that strain is half or a partly strain. Okay, that's why the DNA replication is called it as a semi-conservative. Understood? That means semi-conservative means the new strain which was synthesized, that strain is synthesized from the old strain, and that strain is partly strained from the original one. Okay, and that semi-conservative, that means the DNA replication is a semi-conservative, was first explained by two scientists. Messelson and Stahel. Okay, so DNA replication is a semi-conservative was first explained by Messelson and Stahel. And remember, after the replication of DNA, the new strain which gets synthesized, that strain is always called it as a conservative. Okay, conservative means that is has been synthesized from the old strain, right? This background news really disturbs me. And semi means half, partly. It was first explained by Messelson and Stahel. It is also important. Sometimes the question asks for 2 mark or for 3 mark. 3 marks, most probably 3 marks for 3 marks. Okay, so we are going to talk about the experiment performed by Messelson and Stahel to prove that DNA replication is a semi-conservative. So these two scientists, they have used a bacteria, E. coli. Okay, they have used a bacteria E. coli to perform this experiment. You know, E. coli is a very important bacteria. In every biological studies or biotechnological processes, E. coli always plays a very important role. Okay, any scientist who ever do any experiment, they always used E. coli bacteria. So you know, bacteria, any bacteria, not only E. coli, bacteria divides every after 20 minutes. Okay, bacteria reproduce every after 20 minutes. How fast they are. So these two scientists use E. coli and they have used heavy nitrogen N15. I hope you all already have some idea about heavy nitrogen, heavy element, light element, radioactive element, radioisotopes, from chemistry point of view, you have already discussed, or you have already got all those terms, right? So there are some elements which are heavy elements, okay, like nitrogen N15. They are heavy elements, okay. So they have used E. coli and heavy element. This is not a radioactive element, okay. This is heavy element N15 in the form of ammonium chloride, NH4Cl, in the form of NH4Cl, okay. So they have used two things, E. coli and heavy nitrogen in the form of NH4Cl, that is heavy nitrogen. So next what they did, they allow E. coli to divide, that means to reproduce, with N15 in a medium, okay. In a medium they allow E. coli to divide with heavy nitrogen N15. That means they mix, in a simple word let me tell you, they have mixed heavy nitrogen with bacteria E. coli, okay, or they have injected heavy nitrogen in bacteria body and they allow them bacteria to divide, that means to reproduce. So when bacteria inject N15, that is heavy nitrogen, of course their body will contain, that means their DNA will contain N15, okay. So what they did, let me write it here. They allow E. coli to divide along with heavy nitrogen N15 in the form of NH4Cl, okay. They allow E. coli to divide in the form of, okay, E. coli to divide with NH4Cl, that means heavy nitrogen N15. So of course when DNA contains heavy nitrogen, all the DNA of bacteria will have heavy nitrogen, okay. Now this experiment was performed in a density gradient. Messelson and Stahel, they performed his experiment on a density gradient using a chemical cesium chloride, okay, of this cesium chloride. This background sound, it really distracts me, but still we have to carry on. So DNA replication is a semi-conservative, was first said by or explained by Stahel and Messelson by using E. coli and a heavy nitrogen N15 in the form of NH4Cl. So they allowed E. coli to divide along with NH4Cl, that means heavy nitrogen N15, in a medium, containing in a medium, okay. So definitely all the DNA present inside the bacterial body, they will all contain heavy nitrogen along with it, because they have mixed up N15 along with the DNA or along with the bacteria. And all this experiment they have performed in a density gradient by using cesium chloride, okay. So these are the things that you need to remember. When you write your notes, you can write your sentence. Here, I can give you the exact, I can detect you also, or I can only explain it here. So once you understand, you can write by your own. Definitely you can write by your own. Use your own language, but there should be all those terms in your notes. Okay? Try to build your note yourself. That will be very helpful for you, and it will be very effective for you as well to study. So they allow them to grow along with the N15, then perform experimental density gradient using cesium chloride. Now we need to see what happened. That means density gradient means by using a test tube, they have applied cesium chloride inside it, okay, and then they have applied the bacteria along with the heavy nitrogen. So let me draw the DNA of the bacteria. Okay, after reproducing their DNA, the bacteria of DNA will contain heavy nitrogen along with it. Okay, so heavy nitrogen we write N15 and 15. Let me write this one also. Okay, N15 and N15, that is the heavy nitrogen. This is the bacterial DNA. Then they again allow them to divide, and we know that every bacteria divides after 20 minutes. Now they allow them to divide along with the normal nitrogen, along with the normal nitrogen. The normal nitrogen we write N14, okay, N14 in the form of NH4Cl. Here is N14. Okay, I didn't mention this one before. You can add this point also. So after reproducing the E. coli with N15, all the DNA of E. coli will contain N15 along with it. Okay. Now in his experiment, they again allow those N15 DNA, that means the N15 containing DNA of bacteria, to reproduce again using normal nitrogen N14 in the form of NH4Cl. Okay, that is the normal nitrogen. So next what they have found that, okay, these two strain, the DNA strain of bacteria, they separates. Okay, they separate. This is the original strain, this one here. It contains N15, and this strain, they got separate from each other. Okay, they got separate from each other. Okay, I need a space, so let me draw it that side. I need a space there, otherwise you will get confused. So let me draw here. Okay, let me draw from this part. So here the DNA of bacteria contain N15 and N15, all right. So they allow them to divide using a normal nitrogen, that is N14 in the form of NH4Cl, that is the normal nitrogen. And we know the DNA divides every after 20 minutes. Okay, now what they found out that these two strands, they got separate from each other. These two strains of DNA of bacteria containing N15, they got separate from each other, and they synthesize new DNA. Each strain of bacterial DNA containing N15, they synthesize new DNA. That means they replicate its DNA containing N14. That means normal nitrogen. Okay, try to understand. Okay, try to understand. First, in first case, they have used N15 only. So the first generation, they have found all the DNA contains N15. Next, they allow them to reproduce again with normal nitrogen. They have found out that each strain synthesizes a new strain. That means each strain replicates new strain, but that strain will contain N14. That is the normal DNA, because they have applied the normal nitrogen. Okay, not normal DNA, normal nitrogen. So the N15 containing DNA now will synthesize the new strand, the new DNA strand containing N14. That is the normal nitrogen. Okay. So this strand also, they will synthesize new strand containing N14. Yes. Okay. So that means from the old strand, a new strand has been synthesized. That is the conservative. Simple, simple. The experiment is very simple. They have just used heavy nitrogen and some chemicals. That's it. Okay. Just to see that the new strand which synthesized are from old strand, or they are something separately strand that has been synthesized without using any other old strand. So to see that only, again they have allow those strand to replicate again, to reproduce again. They have, they allow again to reproduce the DNA containing those two strands using normal nitrogen in the form of NH4Cl. Okay, NH4Cl. Okay, this was our heavy strand. Okay, because both strand contain N15. Now in this part, this DNA strand has one heavy and one normal. That means one light. Normal is the light we consider this as a light. Okay, it contains one heavy and one light, or one normal. So this strand, this also similar. So this strand we call it as a hybrid strand. Okay, hybrid strand. Now next, again they allow them to reproduce containing hybrid strand. Hybrid strand means one heavy and one light, or one normal. Again, these two strands will synthesize is one DNA. That means these two strands will separate and synthesize is one DNA. This strand also do the same. These two strands will separate and synthesize is one DNA containing N14. Okay, so let me draw first black in color here. That is our heavy strand DNA. Now the heavy strand DNA will synthesize is one DNA containing N14. You will understand, don't worry. Okay, this is this strand. It replicates is one DNA. Now this DNA will contain N14 because these scientists have applied N14 here. Okay, now here in blue in color, this strand will also synthesize is one DNA. That means this strand will also replicate is one DNA. So here the green in color, not green, blue. Okay, this strand, that is the normal nitrogen containing DNA strand, will also synthesize is one DNA, but we are using normal nitrogen, so they will contain, the strand will contain normal nitrogen, right? This will also do the same. I know you have understood already because you are all smart students, right? or no? I assume you are all smart student. So this strand, the black in color, will also synthesize. It's one DNA, but we are using normal nitrogen, so that the strand will contain normal nitrogen. See, we are drawing a DNA strand in straight form, but it is not as so. Okay, to look it easy, to make us understand, we have to solid it. Otherwise, we have to make it coil structure. No need to do it coil structure. It will look very dirty. So it will do to make us easy to understand. বুজি পাইছা? So এনেকুৱা হয় আৰু কি হয়? ইয়াত চোৱা এটো দুটা যোনটো স্ট্ৰেন আছে এটা স্ট্ৰেনত হেভি নাইট্ৰজেন আছে এটা স্ট্ৰেনত লাইট নাইট্ৰজেন মানে নরমাল নাইট্ৰজেন দিয়া হৈছে যেটা ৰেপ্লিকেট কৰিব দুটা স্ট্ৰেন আমি ৰেপ্লিকেশন অলৰেডি পঢ়িলো এটো যোনটো স্ট্ৰেন আছে N15 এটো নতুন এটা নিজৰ স্ট্ৰেন বনাব মানে ৰেপ্লিকেট কৰিব আৰু DNA N14 স্ট্ৰেনো নতুন এটা ৰেপ্লিকেট কৰিব নিজৰ DNA কিন্তু আমি কি এপ্লাই কৰিছো নরমাল নাইট্ৰজেন এপ্লাই কৰিছো এই কাৰণে যোনটো N15 DNA স্ট্ৰেন আছে যেতিয়া নতুন স্ট্ৰেন ৰেপ্লিকেট কৰিব হেতু স্ট্ৰেনত কি পোৱা যাব N14 পোৱা যাব ঠিক আছে কিউ নহয় কিউ মানে আমি N14 ইউজ কৰি আছো ইয়াত ওকে তো হেতু সেম হ'ব N14 আছে যেটা নতুন স্ট্ৰেন বনাব নতুন স্ট্ৰেনত আমি কি পোৱা যাব N14 এ পোৱা যাব ইয়াৰ ক্ষেত্ৰটো একে হ'ব ইয়াৰ ক্ষেত্ৰটো একে হ'ব ওকে সো আমি নাম দিউ যোনটো DNA স্ট্ৰেন আছে ইয়াৰ এটা DNA হদাই ডাবল স্ট্ৰেনে হয়নে ডাবল স্ট্ৰেনে থাকে এনিমেলৰ ক্ষেত্ৰত হওক প্লান্টৰ ক্ষেত্ৰত হওক মাইক্ৰো অৰ্গানিজমৰ ক্ষেত্ৰত হওক ডাবল স্ট্ৰেনে থাকে DNA কয়েল ফৰ্মত খালি শেপটো অলপ বেলেগে হয় আমাৰ ক্ষেত্ৰত লিনিয়াৰ শেপ হয় DNA এর ক্ষেত্ৰত এ সার্কুলার নিচিনা শেপ হয় আৰু ওকে DNA এর ক্ষেত্ৰত সরি মাইক্ৰো অৰ্গানিজমৰ ক্ষেত্ৰত এ কিবা ওলাই আছে মোৰ মুখৰ পৰা আজি মাইক্ৰো অৰ্গানিজমৰ ক্ষেত্ৰত DNA শেপবোৰ এনেকুৱা থাকে দেই in case of microorganism let me speak a little bit Assamese today okay so DNA এর ক্ষেত্ৰত আকৌ DNA মাইক্ৰো অৰ্গানিজমৰ ক্ষেত্ৰত তো DNA এর যোনটো শেপ থাকে এ সার্কুলার শেপ বুলি কও আমি এনেকুৱা থাকে দেই কিন্তু এনিমেলৰ ক্ষেত্ৰত হওক বা হিউম্যানৰ ক্ষেত্ৰত হওক DNA বোৰ এনেকুৱা থাকে লিনিয়াৰ ফৰ্মত থাকে আৰু সদায় ডাবল স্ট্ৰেনে থাকিব RNA হল সিঙ্গল স্ট্ৰেন ওকে তো হেতু যোনটো DNA আছে আমি যোনটো ফার্স্ট জেনারেশনত DNA পাইছো এটা স্ট্ৰেনত আছে হেভি নাইট্ৰজেন এটা স্ট্ৰেনত আছে লাইট নাইট্ৰজেন তাক আমি হাইব্রিড বুলি কওঁ দেই হাইব্রিড স্ট্ৰেন হাইব্রিড স্ট্রেন ঠিক আছে যোনটো নেক্সট DNA স্ট্রেন আছে আমাৰ দুটা স্ট্রেনতো দুটা লাইট নাইট্রোজেন আছে মানে নরমাল নাইট্রোজেন নরমাল নাইট্রোজেনক আমি লাইট নাইট্রোজেন বুলিও কওঁ দেই তো ইয়াক আমি লাইট স্ট্রেন বুলি কওঁ ইয়াৰ ক্ষেত্ৰতো একে সো আমি লাইট স্ট্রেন বুলি কওঁ হেতু ক্ষেত্ৰত আমি কি কম এটা স্ট্রেনত আছে হেভি এটা স্ট্রেনত আছে লাইট তো আমি ইয়াক hybrid বুলি কম দেই hybrid strain I will speak Assamese today okay তো কি কৰিলে DNA replication is a semi conservative কোৱা হৈছে আমাৰ যোনটো DNA replication হয় তাক আমি semi conservative বুলি কম semi conservative মানে যোনটো নতুন strain এটা বনিব হেতু strain এটা partly বনোৱা হয় মানে completely নহয় মানে partly আৰু এটো স্ট্ৰেন পুৰণা যোনটো স্ট্ৰেন হয় তাৰ পৰা বনোৱা হয় হেতু কাৰণে আমি কনজারভেটিভ কোৱা হয় তাক তো এটো প্ৰথম কোনে এক্সপ্লেইন কৰিছিল দুটা সাইন্টিস্টে এক্সপ্লেইন কৰিছিল স্টেহাল এণ্ড মেসেলসন কি কৰিছিল এটা ব্যাকটেৰিয়া লোৱা হৈছিল ই কলাই ব্যাকটেৰিয়া বুলি যোনটো ইচেৰিচিয়া কলাই বুলি কওঁ আমি বায়োটেকনলজি পঢ়িম এটো ব্যাকটেৰিয়াৰ বিষয় তো ই কলাই লোৱা হৈছে আৰু তাৰ লগত হেভি নাইট্ৰজেন লোৱা হৈছে N15 ওকে আমি হেভি নাইট্ৰজেন বুলি কওঁ আৰু N15 তোমালোকে জানা চাগে কেমিষ্ট্ৰিত পাইছা হেভি নাইট্ৰজেন লাইট হেভি এলিমেন্টস লাইট এলিমেন্ট পেৰিয়ডিক টেবুলত আছে তাত গুটেখিনি তো কি কৰিলে প্ৰথম দুটা সাইন্টিস্টে মেচেলসন আৰু ষ্টেহলে যোনটো ব্যাকটেৰিয়া আছিল ই কলাই ব্যাকটেৰিয়া তাক ৰিপ্ৰডিউস কৰিব দিলে দেই ৰিপ্ৰডিউস কৰিব দিলে কাৰ লগত N15 ৰ লগত ৰিপ্ৰডিউস কৰিব দিলে N15 হল হেভি নাইট্ৰজেন তো যোনটো জেনাৰেশন পোৱা গল তাতে দেখা পালে দে দুটা স্ট্ৰেন ডিএনএ যোনটো দুটা স্ট্ৰেন আছে দুটা স্ট্ৰেনৰ ক্ষেত্ৰতো N15 পোৱা গৈছে তাৰ মানে হেভি নাইট্ৰজেন পোৱা গৈছে হে কাৰণে এটো স্ট্ৰেনক আমি হেভি স্ট্ৰেন বুলি কওঁ ওকে তাৰ পিছত কি কৰা হল নেক্সট আকৌ এই দুটা স্ট্ৰেনক ৰিপ্ৰডিউস কৰিব দিলে তাৰ লগত কি এড কৰিলে কিন্তু N14 এড কৰিলে so N14 means light nitrogen or normal nitrogen okay so next কি পোৱা গল যোনটো ফার্স্ট স্ট্ৰেন আছে তাত এটা স্ট্ৰেনত এটা হেভি এটা লাইট নেক্সট স্ট্ৰেনত তো এটা হেভি এটা লাইট okay so হে কাৰণে এটো স্ট্ৰেনক আমি হেভি স্ট্ৰেন বুলি কওঁ so similarly এনেকুৱা কৰা হল তো next now this experiment he performed using a density gradient okay density gradient using a chemical cesium chloride okay we have already seen that so in a test tube they have used cesium chloride okay along with the DNA strand first this strand we are talking about this strand now okay so they have applied cesium chloride using a DNA strand and they have found out that the DNA strand are at the bottom of the test tube because they are heavy strand okay similarly for this two strands are similar so I will show you one diagram only so by using cesium chloride to check that whether the strands are heavy light or hybrid so they have used cesium chloride using a density gradient and they have found out that this strand just slightly above the bottom part of the test tube okay here somewhere here just below the middle part slightly above the bottom of the test tube so from there it indicates that those strands are hybrid strand now for this so hybrid we have already seen so let me show you light one only so here in this strand they have found out that these two strands remain at the top part and these two strands that is hybrid it remain just slightly below the middle region okay so we have light strand and we have hybrid strand okay so that is why this experiment is called it as a semi-conservative means that is new strand which has been synthesized the strand has been synthesized from the old strand and that is a partly form of that strand okay so we call it a DNA replication is a semi-conservative and this was explained by Messelson and Stahel using E. coli bacteria and heavy nitrogen and light nitrogen as well and cesium chloride as a density gradient okay first of all they allow the E. coli bacteria to reproduce along with the heavy nitrogen the first generation they have found out that all the DNA that contains are along with the heavy nitrogen so that is the heavy strand okay the next generation they have allowed to reproduce again containing heavy nitrogen along with the light nitrogen or normal nitrogen is N14 in the form of NH4Cl So each strand will synthesize or replicate its one DNA strand, but here we are using normal strand. So this strand will synthesize new strand, but this strand will contain normal nitrogen. Okay, so similarly this strand will also contain the normal nitrogen along with that, and again this generation it again allow them to reproduce containing normal nitrogen. So this is the result we find, two light and two hybrid strand. So that means the DNA which has been synthesized, that means the new strand, new DNA strand which has been synthesized are from the old strand. Okay, and it is a partly form of that strand. That's why DNA replication is called it as a semi-conservative. And this experiment was done by using a density gradient using cesium chloride, okay, in a test tube. Okay, so I hope you understood. So this is all about DNA replication process. And next we will see genetic code.